The gut microbiome is an incredibly important part of human health. Bifidobacteria is focused on the study because of its abundance in the gut microbiome and importance in human health, being correlated with positive impacts. All bacteria have lipoproteins on them, which perform vital functions such as nutrient uptake. Bacterial lipoproteins, not to be confused with human lipoproteins, are stimulants of the immune system causing for inflammation. If you look in this image, we can see that within the intestine there is the epithelial lining, which commonly interacts with the gut bacteria and intestinal molecules. And different bacteria can have different structures of lipoprotein on them, such as diacyl, anacyl, or lyso, which have two acyl chains and interact with the TLR2 and 6, or triacyl, which has three acyl chains and only interacts with TLR1. And these acyl chains, if we look all the way on the right, are those squiggly lines on the molecule. And depending on the structure, it impacts the amount of stimulation it has, as we can see through the thickness of the arrow. And understanding the composition of the gut microbiota through the lipoproteins is important because it's an understudied area for a molecule that's commonly found within the gut that is inflammatory, and so can provide insight into when the gut is unbalanced. This study focused on 12 different strains and that range across 5 different species, and there were similarities between the species, as we can see through the longums, which are very similar, and the brevs, which are similar except for brev 2, and in the ungulatum, dentium, and adolescentis being more scattered around. We used two different approaches, one that used the whole cell and the next one that used only the lipoprotein. In the former of the methods, we use human embryonic kidney cells, which are pre-engineered to express toll-like receptors. In an experiment that used TLR2, 1, and 6, we can see that it'll interact with all different types of lipoproteins, regardless of if they are triacyl or diacyl. And we can see that there is overlap between them, but there's also a couple of ones that are not overlapping, such as the ungulatum and longum. By using only TLR21 or 26, we can see which pathway they use, and we can see that in the EC40s, they only interact with TLR26, indicating that they are diacyl and not triacyl, since they do not interact with TLR1, 2 and 1. And we can also eliminate lyso since they have a different EC40 pattern than the positive control of positive control of Enterococcus, which has a lysoform and is a lot higher EC40, which is the amount of cells needed for 40% of the total concentration. And again, we can see that there's similarity between the species, where all the longums, the blue ones on the left, are a lot higher VC40s compared to the brevs, which are a lot lower, but within their own species are pretty much close. And the ungulatum, dentium, and adolescentis are scattered between the two of them again. And so they also have the similarity to the positive control of Listeria monocytogenes, which is a gram-positive diacyl, suggesting that it may be close, but it really doesn't have a way to definitively say which structure it is. In order to do that, we use our second method, which is mass spectrometry. By using purified lipoproteins on an SDS page gel, we were able to separate based on size, and we can identify two bands of interest, one at 40 kilodaltons and one at 50 to 70 kilodaltons. And so when we fragment that and then apply mass spectrometry, we can compare that to expected lipoproteins and create candidate lipoprotein structures. And so these are based on literature findings for the carbon length, and then can be used for further mass spectrometry to fragment it further and be able to determine exactly what structure it has. So to recap all of that, we have the HEC-TLR assays, which were able to identify that they are not lyso or triacyl, and it still showed the similarity between the strain levels, but also showed the difference of a threefold difference between the brevs and the longums. And so in further analysis, we will be using a high and low stimulating strain to be able to perform mass spectrometry and determine whether or not that difference is from the abundance of lipoproteins or the structure of lipoproteins. Additionally, the purification process will be optimized in order to yield distinct bands in each lane, which is something that was not seen in Bifidobacterium adolescentis. And then the impact that environmental fatty acids have on the lipoprotein length will also be studied.